This is part 2 of the presentation on the blood. The third main type of cell that is in the blood are called thrombocytes or platelets. And these are actually fragments of a larger cell called a megakaryocyte that ruptures and releases all of these little fragments. And of course the main function of platelets is to form a blood clot. What you see here on the left, this is a megakaryocyte, and on the right, these are the platelets. Forming a blood clot is a process that normally occurs within about 3 to 6 minutes when a blood vessel has been injured. So the injury of the blood vessel itself causes the muscles in that blood vessel to contract. And so when the blood vessel gets narrower, then that limits the loss of blood right away. So that's an intrinsic reaction of those blood vessels. However, then we need to form a patch over that hole. And so the platelets that are flowing along in the blood start to stick to that site of injury, and they spread along the surface of the blood vessel to help stop the bleeding. At the same time as this is happening, there are chemical signals that are released from small sacs that are inside the platelet, and that attract other cells to come to the area and cause them to clump together, and we call this a platelet plug. On the surface of these activated platelets, there's a series of chemical reactions that occur that involve a number of clotting factors. And this is a whole cascade of reactions that requires the presence of at least 12 different clotting factors. And if you're missing one of those clotting factors, then this, just like a series of dominoes that is missing a domino where there's a big space, then the reactions can't continue and the person can't form a fibrin clot. So this all happens on the surface of these activated platelets, and it requires at least 12 different clotting factors. These are proteins found in the blood. And it causes a whole series, a cascade of chemical reactions. One happens which causes the next, which causes the next, and so on. And a fibrin clot is formed. And again, as I said, if you're missing one of these proteins, if you're missing one of those 12 clotting factors, then you cannot form this fibrin clot. The fibrin forms like a net or a meshwork that traps red blood cells and prevents them from being lost outside the vessel. And that's what you can see on the left side. You can see this fibrin meshwork, and you can see red blood cells that are being caught inside that little net. Within about half an hour, the platelets then contract. It pulls that mesh really, really tight, squeezes any of the water out, and that's what forms a scab. And that happens whether it's internally or externally. It's very dry. Internally, of course, that scab would be quite moist. And that is the patch. And it will allow, as new cells are regenerated, they will replace that patch, and eventually we won't need that patch anymore. Hemophilia is a disorder in which a person can't create a blood clot. And this is an X-linked recessive disorder. And because it's X-linked, it's a recessive gene found on the X chromosome. It's more common in men than in women, and males than females. There are different types of hemophilia, and they depend on which of the 12 clotting factors a person is missing. So for example, hemophilia A is caused by the lack of clotting factor 8, and hemophilia B caused by the lack of clotting factor 9, and so on. In any case, if they can't form that blood clot, then small injuries can cause excessive bleeding. To the point where a nosebleed could be life-threatening. And certainly even small bumps can cause very, very large bruises because blood flow continues, and it's causing more swelling and blood into a particular area. Bleeding that incurs into joints can cause pain and can cause structural changes in those joints too. A person that has hemophilia needs that clotting factor, and they can get that clotting factor from a plasma donation. So if somebody donates their plasma, then they're donating their clotting factors, which are dissolved in that plasma as well. A number of years ago, the blood supply was contaminated with HIV, and so many people with hemophilia that were receiving these infusions of donated plasma then became ill with HIV and AIDS. And so they have done some advancements since then, and now they can either purify a clotting factor from plasma, and they can take that either from human donors, or they can actually genetically engineer clotting factor, which reduces the risk of contamination with something like a virus or bacteria into that from that donation. I would like you to look up in your textbook what is a complete blood count, and it is abbreviated. What is a CBC? If somebody orders a CBC, or a complete blood count, what does that entail? The last section that we're going to do, are about human blood groups. If we look on the outside of red blood cells, the cells are covered with proteins. And we call these proteins antigens because if we put those proteins into somebody else's blood, that person could have an immune response or an allergic type of response to those proteins. So that's what an antigen is. It's a protein that could cause an immune response. So if we look on the outside of red blood cells, there are 30 different antigens that could be there, 30 different proteins that are on the outside, and we have names for them. Not everybody's red blood cells contains all 30. So some people have some of those 30, other people may have a different set of those 30, and so on. There are 3 of those proteins that we want to mostly be concerned with when we talk about how we classify blood. And they are antigen A, antigen B, and a group of antigens collectively that we call the Rh antigen. And these 3 antigens form the basis of our ABO blood groups, or blood typing. So your ABO blood group that you belong to is based on which of 2 antigens, A or B, that you have inherited from your parents. So if you have type A blood, it means that your red blood cells have antigen A on them. If your red blood cells have antigen B on them, then we say you have blood type B. If your red blood cells have both antigen A and antigen B on the outside of them, we would say you have type AB blood. And if you don't have either A or B, we say you have O, type O blood. The Rh factor is expressed as positive or negative. So if you have the Rh antigen on the outside of your red blood cells, we say you are Rh positive, meaning you're positive for that antigen. If your red blood cells don't have that Rh antigen, then we say you are Rh negative. So here's a little chart that summarizes that. So here is a person whose red blood cells have antigen A, but nothing else. They're missing Rh, so we would say they are A and then negative for Rh. On the other hand, if they have the A antigen and the Rh antigen, then we say they are A and then positive for Rh, and so on. If you have A, B, and Rh, all of them, then we say you are AB positive. And if you have none of them, you don't have A or B or Rh, then you are O negative. In order to understand who could donate blood to who, or who could receive blood from who, so which blood types are compatible, we have to understand not only what antigens are on the outside of a person's red blood cells, but also then what part of their immune system is contained in plasma. So for example, if you are A positive, as in this particular case here, then you would have antigen A on the outside of that red blood cell, and you would have an Rh on the outside of that red blood cell. But you would not, in your plasma, have antibodies that would destroy those. That wouldn't make sense. Otherwise, you would destroy your own red blood cells. But you would contain an antibody that against an antigen that you don't have. So, for example, in this particular case, antigen B is not on the outside of your red blood cells. So in your plasma, you would have an antibody against antigen B. And that antibody is there just in case you were invaded, in quotation marks, by antigen B. Then your antibodies would be able to destroy that foreign protein. So we contain in our plasma antibodies that will bind to any antigens that our body considers foreign, meaning that they're not part of ourself. If you were exposed to a protein, so in this particular case, if you received blood that contained the B antigen, so this particular person that doesn't have a B antigen on their red blood cells, so they're going to have antibodies against that. If they received blood that contained a B antigen protein, then their antibodies would bind to them, and that would cause a reaction. And the reaction is called agglutination. And this reaction, this binding of antibody and antigen together, causes red blood cells to clump together. And this is very dangerous because this clumping can clog those small blood vessels, which means that oxygen can't, or blood and oxygen can't get to tissues. It can cause red blood cells to be ruptured, which causes severe anemia, and it can cause kidney failure as well. So the A, B, and Rh antigens, out of all the 30 different possible antigens that are on the outside of red blood cells, A, B, and Rh cause the most serious reactions. And that's why we type blood based on these 3 out of the potential 30, these 3, A, B, and Rh. Those are the ones we use to type a person's blood. So this is very important, that a donor's blood cannot contain any red blood cells that will be attacked by the recipient's antibodies. Just an interesting and important note to this, and that is that antibodies against antigen A and B are present at birth. However, Rh antibodies are not present in the blood at birth. So if a person that is Rh negative receives Rh positive blood, then they will produce anti-Rh antibodies. So it means the first time that they receive Rh positive blood, they don't have a reaction, but they will then produce those antibodies. And the second exposure to Rh positive blood will then produce this clumping reaction. For the purposes of receiving and donating blood, we never take that risk. We never say, oh, maybe this person has never been exposed before to that antigen. So we always assume that there will be a clumping reaction. I would like you to look up in your textbook and answer the question about what is erythroblastosis fetalis. Sounds like a big name, but it really is about why an Rh negative mother maybe have some difficulties with an Rh positive baby, and the difficulties are with the baby. So if we want to determine the blood type of an individual, then what we can do is we can take an unknown sample and we can add known antibodies. So for example, we could add antibodies against A, antibodies against B, or antibodies against Rh. If we see a clumping reaction, then we know that that particular antigen is present. So for example, in this sample right here, let's say that we took that sample and we added anti-A, and we saw that there was no clumping reaction. Now we know that antigen A is not there, and that person cannot be a blood type with an A in it. However, if we add, and let's say in this case we added anti-B and we see a reaction, now we know that B must be present. So that person must be either blood type B. It can't be AB because there was no reaction to A. We can do the same thing by adding anti-RH to find out if they are RH positive or RH negative. Here's another example. So here, if we add anti-A serum to this sample, we see no clumping reaction. So now we know that this particular person does not have antigen A. So could not be A or could not be AB. Then we add anti-B to this serum to the sample. No clumping reaction occurs. That means that antigen B is also not present. So we know they're not A, they're not B, and they're not AB. So that leaves O. So now we know they are blood type O. The last thing that we need to do is find out, are they positive or negative for RH? So here we add the RH, anti-RH, and we see a clumping reaction that occurs. So that means that this person's red blood cells have antigen RH, meaning they're positive for RH. And so now we know they are O positive. So that's how we would figure out an unknown sample of blood to what type that was. Here's a question. So here, anti-A was added to this sample. This is all the same from the same person, and we're going to test to see whether or not they are A, B, or O, and whether they're positive or negative for RH. So in A, we added anti-A, and we saw no clumping reaction. In here, we added anti-B, and we saw clumping. And here we added anti-RH and we see clumping. So what blood type are they? And they are, in fact, hopefully you got this answer right, they are number 2, B positive. How about this one? Take a moment to figure this one out. If you said O negative, number 5, then you'd be correct. No clumping to anti-A, no clumping to anti-B, and no clumping to anti-RH, meaning they don't have any of those antigens. A person who is O negative is called a universal donor, and that's because they have no antigens on their red blood cells, they can go into anybody's blood and they're not going to have a reaction with whatever antibodies are in that person's blood. So they can donate to anybody. The flip side of that is that they can only receive O negative blood because their blood is going to contain antibodies to all of the antigens. Okay, how about this one? If you said number 3, AB positive, then you would be correct. So we see clumping in all 3 of the samples, to anti-A, anti-B, and to RH. And so that means that there are antigens A, B, and RH present, so AB positive. Somebody who is AB positive has no antibodies in their plasma, which means that they can receive blood from anyone. So we call somebody who is AB positive a universal recipient. Here's a question: which blood type is the most common in the population? The answer is number 1, O positive. So about 37.4% of the population has O positive blood. A close second would be A positive, where 35.8% of the population has A positive blood. Which blood type do you think is the least common in the population? And the answer is AB negative. So only about 0.6% of the population is AB negative. Other very uncommon blood types would be B negative, only 1.5%, and AB positive, 3.4%, followed by A negative, 6.3%, O negative, 6.6%, and B positive at 8.5%. Okay, this ends the presentation on the blood. There is a $50 HPH dollar opportunity that is posted on Blackboard. It is in the HPH dollars section, and there's a folder in which there's some background information as well as a connection to a link to a quiz and a fun game to practice your knowledge of human blood groups. This is the end of the presentation, and you will need this information for your online test and midterm test. Thanks very much.